So thank you all for joining us for Café Sci this evening. My name is Brad Peroni. I will be your host. And our speaker tonight is Dr. Tullia Bruno from University of Pittsburgh. We have, of course, our sponsors to thank this evening. Café Sci is presented by PPG and sponsored by Cook Myocyte. You can catch Café Sci the first Monday of each month here on Zoom webinars. You can find all the information for future Café Sci's at our website, carnegiescienceCenter.org. Now, it's my pleasure to introduce Dr. Tullia Bruno. Dr. Bruno is an assistant professor in the Department of Immunology at the University of Pittsburgh and a faculty member in the Tumor Microenvironment Center and the Cancer Immunology and Immunotherapy Program at the UPMC Hillman Cancer Center. She obtained her PhD in immunology from Johns Hopkins in 2010 and completed her postdoctoral fellowship at the University of Colorado in 2015, both with a focus in tumor immunology. While Dr. Bruno's PhD training focused on inhibitory receptors on intratumoral T cells, she became interested in the role of B cells in the tumor microenvironment during her postdoctoral fellowship and has built her independent research program around understanding intratumoral B cell function in multiple human cancers, in particular lung and head and neck cancer. Dr. Bruno's research lab has an overt focus on studying immunity within cancer patients, which makes her research highly translational with the potential for future clinical trials targeting B cells. Thus, Dr. Bruno's overall research objective is to develop a B cell specific immunotherapy in the next 5 to 10 years. In addition to her work in cancer, Dr. Bruno has been co-leading efforts at the University of Pittsburgh to study the immune response in local individuals that have been infected with COVID-19. With that, it is my distinct honor and professional pleasure to introduce Dr. Tullia Bruno. Welcome, Tullia.
Hi there. Sorry that my lighting's a little bit not great. I'm with my parents this evening because I just picked up a new vehicle in Erie. So I'm using one of the spare bedrooms for this, but it doesn't really matter because I'm gonna be with my camera off most of the time just for the sake of presenting. So just as was mentioned, make sure to use the Q&A function when you have questions. And definitely I'll have some pauses for questions throughout the presentation. I'm gonna go ahead and start sharing my slides, and I'm going to stop my video. Okay, so as been mentioned, first of all, I want to thank everybody for being here tonight. I can see the turnout. It's great. It's great that people want to learn about the immune system. So as was mentioned, I am an immunologist by training, and I'm going to talk about immunity. And I've been told that this is not something that you have been talking about at other Café Sci's. So I thought it was a good opportunity to talk about the immune system, especially given what's going on with the pandemic. So I have a few things here. I have my email address. So if there's other questions that we don't get to tonight, please feel free to reach out to me, but definitely give me some time to respond because there's one of me and looks like there's almost 70 of you logged on right now. And I'm going to be focused on the immune response and what the immune response means in relationship to vaccines and why vaccines are important. I thought that was a timely topic given what's going on and that people are getting vaccinated currently. But I also want to, if we have time, talk a little bit about cancer. It really depends on how many questions you have, and I'm happy to kind of truncate the presentation so that we can take time for questions. So we'll just kind of see how it goes. One thing I want to point out on this slide is that we have here basically an image that was captured from my laboratory. This is an image from a tonsil, so a normal tonsil like you and I would have in our bodies. No disease, no virus, no cancer. And this is what immune cells in those areas look like. And there's these little hubs here that you can see that are called germinal centers. And we're going to talk a bit about how the immune system learns in education tonight. And this is where that happens. This is where the action happens in your lymph node or your tonsil or other lymphoid organs in your body, which I'll point out here in a few slides. So this is something that my lab's become pretty proficient in, which is taking really beautiful images from cancer patient tissues. And now we're starting to do a little bit of COVID work as well, which is ongoing. So I won't show that today, but we can get these beautiful they're called multiplex images of human tissue. Okay, so I thought I would start out actually by just talking a couple minutes about myself, because I think it's important that you know where the person who's presenting came from. So this is my scientific journey. So actually at the age of 6, I wanted to be a waitress because I idolized my grandmother, who actually unfortunately passed away almost 12 years ago now. And she actually passed away to lung cancer. So that was part of the incentive for getting into lung cancer work. And so she was a waitress, so I really wanted to be a waitress. But even at an early age, I had science in my brain, and I was very interested. And at 16, I wanted to be a doctor that saw patients in MD. And that was what I wanted to do. And then that kind of fluctuated between that and a pharmacist. And then I went back to doctor when I went into undergrad. And then I said, okay, well, let me see what else is out there. So I started working in a lab as an undergrad. And I got a really great experience. And I quickly converted and said, I want to get my PhD. And this is what I want to do. So that's when I made the choice to become a scientist. And so I got my PhD at 27. I moved to Colorado and did my postdoctoral fellowship. And then got my first faculty position at 32. And this is myself and my friend, Greg Delgoff. We graduated from Hopkins together, and actually we're both faculty at Pitt together now. So that's pretty fun. So it's come full circle, and now we're back together, which is really fun. I want to also say what I do. So I run a research lab at the Hillman Cancer Center in Shadyside. So really what I'm doing is I'm running a research program, and I teach, but I teach by example and by doing. So PhD students, MD students, high school students, college students that come into the lab and want to learn how to become a scientist. I do research, I generate ideas for experiments and other people in the lab to perform experiments. Occasionally I still do experiments because I am the best trained hands in the lab. But that's not as often as I'd like it to be anymore. I write a lot of grants. I actually just put in a huge grant submission today before all of this. And we often, communicate our research through publication in scientific journals. And also before COVID, we traveled a lot around the world to give talks or go to conferences. And that's really how we network as scientists. And that's really something that's been missing during this pandemic, just the opportunity to go and see our colleagues at these meetings and really exchange ideas, because that's where a lot of these new ideas and grants come about. And then we also do service to the university, such as teaching, et cetera, to help out. This is my mentorship journey. So I started as an undergrad, as I mentioned. I was at Vanderbilt University, and each mentor that I had, primary mentor I had, taught me something. I've had a lot of other mentors along the way, but I don't have time to talk about all of them. And I still have several mentors. As a faculty member, you are constantly learning, which is part of what I love about this profession. But this individual, Daria, he took me into his lab, and he took me in because he said I was very persistent. And that was where I built my foundation. At Hopkins, I was with Chuck Drake, who was an MD, PhD, and he was actually the one that convinced me just to stick with my PhD and not get an MD, even though he had both. He said, you have really good hands, you'll be good at what you do, so just stick with it. And so I did. And so he taught me about controls in experiments, and that there's no such thing as a failed experiment. In fact, you can learn something even if you fail, which is important, because when you're in graduate school, you fail about 90% of the time, and then 10% of the stuff you do works, and you get to publish and pass your thesis. So that was a really good lesson in resilience. As a postdoc at University of Colorado, I really actually learned from Jill Slansky in that postdoc or that training phase how to communicate effectively. And that's really where I found my scientific identity and started working on B cells in cancer, which is a new research niche and something that's really helped me with my career. And then lastly, I have a mentor, a faculty mentor, a primary one at Pitt, and he and I work very closely together on projects, but because he's a very seasoned scientist, he has pushed me to become a better scientist and really think high level, write larger grants, talk about how to manage a lab, which is not the same as mentoring students. You actually have to manage these individuals and funds. And so that's been a learning curve, and his guidance has been super appreciated. So the other thing I just want to mention before I go into the talk is my scientific and personal. So everybody thinks scientists are not sociable and we don't have lives outside of the lab. Partially true, but not 100%. Actually, I waited to kind of focus on my personal, and actually it was just timing. And so I met my husband when I was 29, when I was in Colorado. We moved to Pittsburgh together in 32. When I was 32, our daughter was born 3 years ago, and this is a little outdated, but I'm now 3 years post baby, so my daughter's 3 years old. And this is a picture of my husband and my daughter and I in Halloween when she was 1. She wanted to be an aerobics instructor, so we all dressed as aerobics instructors. And this is her now at 3. So very, very fun to have both scientific and personal, and they definitely balance my life. Okay, so this is my thing about asking questions. If you don't ask, I can't answer it. So I always put this up and say, please ask questions, even if you don't feel like it's a smart question. There's no such thing as a dumb one. You don't learn unless you ask questions, and this is the motto in the lab. So please ask if you don't know the answer, or if you want to know the answer, and I'll try to get to as many as I can today. Okay. So we're going to start with a simulation. This is a simulation of what happens when a virus comes into the community. So the red blocks are infected individuals, and the blue are uninfected individuals. And this is just showing you how quickly a virus spreads if that one individual comes into contact with the 3 around it, and then 3 turns into 5, and then 5 turns into X number, and so on and so forth. And then at some point, everybody's been infected, okay? So this is a very simple simulation. If we were in person, I would have given everybody an index card and we would have done this as an activity, but this is the virtual life. So that's what we have for today. Now, this is the same simulation, but now we have green and red, and green are people that have been vaccinated. So what you'll see very quickly is that the people that are infected still infect those around them, but those in green are not going to infect those around them, and going to, in fact, then cause that infection to be reduced. So this is what is known as herd immunity, and what everybody's been talking about with the COVID-19 vaccine. And it's what happens with the flu vaccine. So we have to get so many people vaccinated in order for the rest of the population to be protected, because if these individuals are vaccinated, they are going to then not spread virus to other individuals. So how do we figure out how quickly a pandemic or an epidemic takes off? This is called the reproduction number, or R0. So R0 basically measures how quickly it will happen. And if it's less than 1, that means they're going to decrease with each step. But if it's greater than 1, which is usually the case, it's going to increase with each step. So it basically measures the spread. And so this is kind of a similar thing what I just showed you with the simulation. You know, if person A is infected, they infect 2 people, then 2 people infect another 2 people, and then those 2 infect 2, and then so on and so forth. So this is what happens with any virus, with a simple cold, with a flu. But with COVID-19, the reason it's so aggressive is because it's just a little bit more of an aggressive strain. It has more severe symptoms. It's got more of a death rate, as we all know. And so that's why when all of this occurred, that's why the CDC said, okay, let's try to put some controls and take care of this transmission the natural way. And this is a simulation showing you what happens in pink with uncontrolled transmission, and then with blue when you have controls. And that's what people are talking about when they say flattening the curve. So it's over time, we try to flatten the curve, and that's the number of concurrent cases. And the reason we try to do this is you can see here, our health care system, as we've seen pretty clearly with COVID-19, comes to capacity quite quickly. And so we want to make sure that we're trying to do everything we can to take precaution. So with COVID-19, this was mandated to stay at home at first in quarantine. And actually we did that because we didn't really know what to do. And I actually would argue that when the virus first came out, we were in much better shape than later, last year, end of last year, when it got really, really aggressive and the cases went up dramatically. And so really it's because the controls that we're putting in place are helping a bit. And so that was keeping people at home. And then we kind of loosened the reins a bit, and then people kind of went nuts. And so then we said, okay, you have to wear masks all the time. And then the face shields are helpful, and, you know, don't touch each other. Don't be in closed spaces 6 feet apart, right? So, and then we had to shut down again because people weren't paying attention, but it really does work if people do it. And we've seen that evidence in other parts of the world, actually. Now with the vaccine, this should start to happen naturally as well. So it just will help it even more. And this is to give you an idea of what happened with the flu season in 2019 and 2020. These are the numbers. So this was the CDC's estimate of how many illnesses, how many hospitalizations, and how many deaths related to the flu. So it's a common misconception that the flu doesn't kill people, but it can. It's just more enhanced with coronavirus. And in fact, you're probably exposed to coronaviruses a lot each fall, and you just don't realize it. And oftentimes you think it's the flu. It's just that this particular coronavirus was more aggressive this time around, and that's where we came into problems. But you can see here that vaccines work. Okay, so if 52% of the US population was vaccinated, which was the target goal for the flu, 6 months, basically over 6 months, okay, you see that those number of cases went down, hospitalizations and deaths were under 10,000. So that's quite good, actually 7,500. So this is why we think vaccines work, and we know that vaccines work. And this is another slide just to show you how herd immunity works, or the concept of herd immunity and the official definition, okay? So again, this is showing you similar to what I showed you in the simulation, right? These are all infected individuals, nobody's vaccinated, but as soon as somebody's vaccinated, it's gonna be protective for other individuals because you're not gonna have disease transmission. And the resistance basically to the spread of infectious disease is basically pre-existing immunity of a high proportion of individuals. So that's why that R naught is important. It tells us basically how many people we think we have to vaccinate in order for things to be effective. Now, the considerations here are twofold for COVID that I'll mention here and we can talk about later. One is that now there are a lot of reports in the news, I'm sure you're seeing, of mutant variants of COVID, and we'll talk a little bit about that later. It does happen, and it can cause shifts in the virus that are then not protected by the vaccine. So that is a real thing. And then the second thing is that vaccines are given systemically, right? So, or intramuscular. So if anybody's gotten their COVID vaccine, and I actually have, so I've gotten both shots of Moderna now. I actually had my last shot last Friday, and it did put me down a bit. We can talk about that, but that's just your immune system working. So it's nothing to be worried about. And basically, you know, when we think about this, you're getting an intramuscular injection. And so when we think about the COVID-19 virus, it is transmitted through the nasal passages, right? And so what happens is we know that the vaccine, if given intramuscular, can make it to the lungs, which is one of the areas that are affected by the virus, and can be a lot of damage to the lungs, as you've probably seen in the media. And we know that we can protect there. What's unclear a little bit still, and the jury's still out, and we're still trying to, you know, do the research on this, is whether or not it makes it to those mucosal areas, which are your nasal passages, effectively. And so this is why somebody like myself, who's been fully vaccinated, still wears a mask every day until it's deemed okay not to. And it's because even if you won't get sick because you've now been vaccinated, if somebody breathes on you that has COVID-19 and it's in your nasal passages, we're not sure exactly how long it can stay there. And it's anywhere from, you know, 4 to 7 days is what people are saying. But basically, it can stay there. And if you breathe on somebody else because you're asymptomatic, then, and they're not vaccinated, then you've now passed it. So that's why we're still wearing masks, because we're still trying to understand the penetrance of that. But overall, if enough people get vaccinated, this is not going to be an issue anymore. So what about the R naught, and how is it basically calculated? So here's the equation, which is less important. What's important are the numbers. It tells you kind of the severity of the disease, right? So flu is a 1 or 2, and that's actually pretty low. And you can see something like measles is a 16 to 18. So it's super, super aggressive, and hence why we have vaccination for such diseases such as that, so we don't have to suffer through them. And also chickenpox is pretty high for kiddos. So, you know, when I was a kid, we didn't get vaccinated against chickenpox. You got it, you established an immune response based off of getting infected, and then you didn't get reinfected again, hopefully. Sometimes it happens and it comes back as shingles, as you all know. Now they vaccinate for chickenpox. So my daughter has gotten a vaccination, so hopefully when she goes to school, she'll be protected. And we'll talk a little bit more about how that works here and with the immune response. So just to give you an idea, COVID-19 is somewhere between a 2 and a 3. And so that's why they've been saying that we have to vaccinate between 50 and 70% of the population in order for the vaccine to work. So this is my soapbox. I will not do it again, but I highly encourage all of you to get vaccinated. There are some really great vaccines. J&J just put out a ton of doses, as you saw in the news this week. And so that's really great. And that's a single shot, so that's even better. But all of them are very effective, and all of them we're doing really well with all of them. So I would highly encourage you to get vaccinated. If you're a little bit hesitant, You could wait a couple more months to see what the new data show on some of these vaccines. But I think from my standpoint, it looks really good. There's been a lot of scientists that have vetted these data. It's been through the ringer with the FDA. So, a lot of this has been really well planned and executed. And the reason it's been, fast tracked so quickly is because we have an active population that's infected. And so it allows us to be able to test the vaccine very quickly and make sure it's working. Okay, so now I'm going to pivot from that background information to the importance of the immune system. And then I'll take a quick break to see if there's any questions that we can answer kind of midway through. So the importance of the immune system. All of you know what your immune system is in general. You know that it's going to protect your body from foreign predators every day. And so that is a range of things, this bacteria, viruses like we're talking about now, even parasites, and in the case of cancer, also cancer. It's a little trickier with cancer because cancer comes from your body, right? It comes from your regular cells and your breast or your lung basically populating and essentially reproducing themselves in an unchecked way. And so it makes it a little bit harder if it's seen as self and not non-self. But if we have time, we'll talk about that today. But in general, we can make immune responses to insults from the outside, basically, and to protect us. The immune response is harbored in multiple places in your body, and this shows you where those areas are. Bone marrow is one area where it really develops. I talked about the tonsil. This is a picture of that just as a reminder. Your lymph nodes are all over your body, and there's a lot of them throughout your body, and this is a really active area of immune system education, which we'll talk about today. Your spleen as well. And some of the other areas that have specialized immune parts. So for example, Peyer's patches in the small intestine are little areas of clusters of immune cells that help regulate what happens in your intestine, for example. And thymus is another place where it's developed. So these are all areas where you can see them, and these are some of the different outside predators that we are protected against. And then the one thing I also want to remind you of is that your skin is the #1 barrier to all of that. It is very protective and is your initial barrier to the outside world. So there's two arms of the immune response. There's the innate and there's the adaptive, and they're called that way. They're called those things for a reason. Innate means it happens right away, and it's something that's there already, and it's going to trigger a response, and it's going to just try to control that response as best it can without any sort of education. So it's usually short term, it responds quickly, but it also is not specific. So what that means is it just generates a robust immune response, but there's not a specificity for that virus because there's no education that's happened to that virus. And so these are some of the cells in your body that are part of your innate immunity. Two of them that we're going to talk about more tonight are the dendritic cell and the macrophage, because these are your cells that are gathering those, virusly infected cells that have died, for example, and taking them up, and they're going to be your educators. They're going to be that bridge to your adaptive immunity. Okay, so some of them function as an innate immune response and try to do that, but then they're also offer that bridge for adaptive immunity. And in the adaptive immune response, there's two things we think about: the cellular immune response, and then also the humoral immune response. So what that means is T cells, which are your actual cells that can target the tumor or the virus, and B cells, which can make antibodies against the virus. And so one thing I want to note here is this is where your memory comes from of your immune system. So if you get a vaccine or you see the COVID-19 virus once, your body's going to remember. And it's going to remember because of that adaptive immunity, because of that education that occurred. And so there's been a lot of, reports in the literature and articles about, oh, well, is COVID-19 immunity long-term? Should we worry about it not being? And the bottom line is if you've been exposed to a foreign particle of some sort, your immune system is going to get educated, and that education is still going to be there, whether you have antibodies in your serum or plasma or systemically present when they test you or not, because your B cells or your T cells can be reinvigorated to make that response again. And so we'll talk about that here in a minute. So this is the same thing, but basically in words, because I appreciate that some people like visual and some people like to read. So basically just the highlights here is that innate immunity is your primary defense. It includes your physical barriers, as I mentioned. So surfactant on your skin itself, your multiple layers of skin are your first line of defense. And then this is going to be mediated non-specifically by cells that I had mentioned in the previous slide, and they produce these proteins, which are called cytokines, that then can help generate a rapid immune response. And it's very rapid temporally as well, 0 to 3 days this occurs. And so it's really that your adaptive immune response takes over if it can't control that infection for 0 to 3 days. And then the other thing that it doesn't have is memory or secondary response. So this is just literally something that's there at that site to help you out in the very direct immediate infection. And a lot of times it can clear it, and we probably get sick less than we should because of innate immunity. So I think it is underappreciated. For adaptive immunity, it's systemic. It protects your entire body, not just at that site, for example, and it's antigen specific. So what that means, the word antigen is the protein that your immune system sees and educates against. So in this case, if it's a virus that's broken down, it becomes an antigen. And so that is going to make it a specific response. So you can actually see those virally infected cells and go after them directly, or you see that cancer cell and you go after it directly. Again, it has memory. It occurs later. It takes about 4 days for this to even start. And it has 2 arms, as I mentioned, both antibody or humoral mediated and cell mediated. So this is a curve showing exactly what I just said. Okay, so this is showing the readout here is antibody concentration. So this is B cells basically that are making antibodies against your virus or cancer, etc. So what happens is here, this is your initial exposure to antigen. So this primary immune response develops over time. So about 4 to 10 days, you get this adaptive immune peak, and then it subsides. So one thing about your immune system, it's very smart. It will clear what it needs to clear, and then it subsides. So that's why when I said, oh, I felt really, not great after my second dose, it's just my immune system remembering what it saw the first time I got my first dose, and then it's going to make that immune response, and it makes you kind of feel not great for like a day or so, and then it goes away. So when you start to feel better, that's when your immune system or your immune response is subsiding and coming back down to baseline. This is the same thing that happens if you get a vaccine. So either if you get infected or you get a vaccine, you have that initial exposure to that virus or that antigen. And what's great about the immune response is that when you have a secondary exposure, like here, you basically have a faster response and a larger response as a result. So therefore, you should not even get sick the second time around. So this is why vaccines are so important, is because it tries to prevent you from feeling sick, and it primes your immune response so that when you see that antigen again, you get a response. And so here's a little graphic just showing all the interplay of all of the immune parts. I thought it was cute because it's a cartoon, but these are all the cells that I told you about. So macrophage is going to be the one that's going to be part of the innate immune system, and it's going to gather these germs, and dendritic cells do the same thing. And then they can educate either CD8 T cells or helper T cells or B cells, and then antibodies can be made and they can mark that germ or that virus and destroy it. So it's a very simplistic view, but it's good if you've never learned about the immune system before. Okay, so this is all why we need a COVID-19 vaccine. This is why we vaccinate in general. I thought that this was good because it just shows the timing, as I mentioned, for innate versus adaptive, and puts all those cells together on a timeline. So if you're going back and watching this again, which you most likely will have to because it's a lot of information in a very short amount of time, you can use this as a learning tool for what I just said, and it intertwines everything that I said about innate and adaptive immunity. Okay, so a couple more things, and then I'll pause to take a few questions. So one of those cell types from the innate immune system that acts as that bridge or that educator is this, what we call an antigen presenting cell. So antigen presenting cells are going to be your macrophages and dendritic cells. And actually B cells do this really well too. And I would be remiss if I didn't say that, because my entire research program talks about B cells and cancer. And one of the things we look at is how they present antigen to T cells. And we know that they can do it, and we've shown that. And so basically that bridge is that this APC, whether it's macrophage, dendritic cell, or B cell, picks up these proteins, these foreign proteins or cancer cells that are dead, and they process those antigens internally. And I won't go through the whole process because it's a long process. And that then causes this bridge with the T cell receptor, or if it's as a B cell, the B cell receptor. And that's where the education occurs. And this is actually a real life view of a T cell and APC interacting, and you can see they're interacting and that education is occurring. So all of this is happening because these antigen presenting cells are very, very efficient at picking up foreign particles and basically getting stimulated and then migrating to where they need to in order to activate and educate a T cell or a B cell response. So what's different about them is that macrophages are local most of the time. So they don't really migrate as well as like a dendritic cell. So macrophages are actually present in almost every tissue of your body, and they're specialized for each tissue. So for example, lung has a specific macrophage that resides there. Your liver has a specific macrophage because they are the eaters of the immune system. So they take up debris, they eat everything around them. And therefore they try to help with that local immune response. And we know that local education is important as well. The primary education often comes from these dendritic cells in the lymph node. So as I mentioned to you, there's multiple sites where we see these very nice round education centers that I showed you in that first slide. And those education centers are going to be where dendritic cells crosstalk with B cells and T cells and educate them to become specific in order to target virus, bacteria, etc. So this is just another slide showing you that exact same thing. So here, the MHC is what we call the protein that your processed viral or bacterial antigen is processed on and loaded onto. And MHC is what actually is going to be matched when you also get a transplant. So when they say, oh, we have to find a match, that's how they match individuals. Okay, so it's a common misconception that if a child needs a transplant, that the parent is the best match. It's possible. It's very highly unlikely, and that's because the MHC encoding for that kiddo came from 50% mom, 50% dad. So it's going to be very highly unlikely that they're a match. Okay. So it's better that it's usually the sibling is a better chance than the parent, for example. And so that's what they're matching, is they're matching your MHC to make sure you don't reject that new organ, okay? So it's the same idea. You're still loading proteins on here so that you can recognize either self or non-self and then educate your T cell. So that's that antigen presentation step. And then the second step is this co-stimulation step, which basically activates the T cell really, really nicely. And then you get proper cytokines from the external, which is signal 3, and it activates them, and then they can go on their way and begin to look for those infected cells or those cancer cells to target them and kill them. So this is a really simplistic view of that entire kind of set of paradigm that I was showing you, from the point of getting this foreign body, into an antigen presenting cell, presenting it on a protein known as MHC that makes you, and then, stimulating the T cell. And then from there, the T cells can be helpers, which I won't talk a lot about today for the sake of time, but they basically do exactly what they say. They help the immune response by either stimulating macrophages or B cells to make antibodies. And this can go back and forth. Actually, B cells can stimulate T cells, CD4 T cells, and then CD8 T cells, which is still a T cell and still educated the exact same way, they become your killer T cells. So they can directly go after, virus or tumor. Now, what I can tell you from here is that I'm giving you a very simplistic overview tonight, but everything is more complicated than it usually seems, right? And so, for example, we're studying now some B cells in cancer patients that we think have the ability to not only, make antibodies, but also present antigen, but also directly kill tumor cells. So that's why this is an active area of research, and why I always tell people that I'll never be out of a job unless I want to be, because you really have so much to learn, and we're ever evolving. So this is the paradigm, and that's what the research is for. So in talking about these killer T cells, before I break for a second and take some questions, the CD8 killer T cells are the ones that a lot of the focus has been on for viral infections as well as for cancer, because they really truly have been shown to be able to directly kill a viral infected cell or a tumor cell. And so this is the same kind of schematic that I showed you, just a different picture of it, where you have this education of the CD8 T cell, and they can make several cytokines such as interferon gamma, which can basically increase its cytotoxic activity. The other nice thing about it is that it can, it recognizes its targets through MHC, okay? So the same thing that educated it, okay, which is here, this little schematic here, is going to be what it recognizes on the other virally infected cells. So it's almost like there's a little flag that goes up, the virus, virally infected cells, and it's the same exact specificity that the CD8 T cell was educated against. So it can leave the lymph node and go and find those virally infected cells and then target them. And this is how it directly does it. So what it literally does is it pokes holes in the virally infected cell with what's known as granzyme. So that makes the little shuttle, and then perforin comes out of the cell and it enters into the virally infected cell, and then it kills it. So that's just one of the ways. There's other ways, but this is the most common way that we usually teach upon. So this is how they actually directly kill. And then the last thing I'll say before I take some questions is that the virus actually can kill, or excuse me, the CD8 T cell, otherwise known as a cytotoxic lymphocyte or a CTL, it can actually kill multiple targets. So that's the nice thing about this, is that once that education happens in the lymph node, migrates out of the lymph node, goes to the site of infection, starts to kill those infected cells, it'll kill each cell until everything is gone. And then the immune response then subsides as a result. Okay, so I'm going to stop here and leave this up while we take some questions. I think it's a good breaking point. This is a summary schematic of what I went through of education and adaptive immunity. And so we can be looking at this while we're taking some questions here. And I can either monitor the chat. Let me look. Okay.
So Tullia,
Wow, sure.
I saw a couple people put their questions into the chat, and they've put them all into the question and answer box. And
Okay.
For those of you who may be pondering some questions, there is a dedicated Q&A tab at the bottom of your Zoom window. So go ahead and click that, and you'll be able to enter your question there. And I believe you'll also be able to see all the questions that people are asking.
Yeah, I can see them. So I can just kind of go down the list and answer. It looks like there's 6 of them right now. So I'll try to do these first and then kind of keep going, and then we'll see what else comes in. So the first one is, how can we combat all the vaccine disinfo and denial in real life and on social media? Oh man, that's a stacked question. So I think it's hard, right? Because it's a lot of misinformation, I think, that's passed around. And I think a lot of people that are just scared of what could happen, especially because the production of the vaccines happened very quickly. And I think that in general makes people nervous. What I can tell you is that what I've done is I've just armed myself with as much information as possible. Like, I don't make vaccines for a living. That's not what I do. I study the immune response. And because I study the immune response, I can say what the immune system does in the context of a vaccine, but I've just been doing a lot of reading, and I think it's finding the right articles. So if you want to arm yourself with some of that, I'm happy. I have like a reading list that I've put together for the OSHA course that I'm teaching now, and I see I have a couple people that are in my class that are here. Welcome, guys. It's good to see you. So I think that arming yourself with the right information is the key, right? And so I've been putting together reading materials for that class to kind of help them, think about all the different things that are out there and kind of mine through the things that are right versus not. Okay, can you explain the role of any of IgA in the control of COVID, and if any options for a nasal vaccine are viable? I don't know the answer to the second one. I wish I did, but I don't. I know that they've been thinking about it, but that's all I know. I think that it's a good thought, quite frankly, because of where it harbors. Those are a little bit trickier than what they're currently giving, so I think that's why they went the route that they did. As far as IgA, it definitely would have a role in the lung, right? So any mucosal site, IgA antibodies are quite important. And so that's gut is the major one people think of, but actually your lungs are another huge one. And also we think about this in the head and neck space because I work on head and neck cancer. And so actually when we measure antibodies for cancer specifically, we measure IgG and IgA because they can have definite effects, and especially mucosal sites. Viral variants occur in different countries. I think that so my best stab at this and kind of just saying out loud, what I'm thinking is that I think that it honestly just has to do with the types of antigenic drift and antigenic shift that we see. And I'll introduce that here in a few minutes. And so you know, even with cancer, some of the mutational burden that we see in patients, it's due to environment as well. Now, that's not necessarily the case for the virus, you know, because it's going to adapt on its own. But I think it's just the type of variants that are there and that are evolving are going to be the ones that are going to be able to also infect people and that are susceptible. And so that's how that starts, right? And in general, does a more severe reaction to vaccine mean that the body is mounting a stronger immune response? If so, what about people who have no adverse reactions to the vaccine? Is their immune response weaker or non-existent? I get this question a lot. No, that does not mean anything if you have a stronger versus not. I can tell you that all of my lab got their second dose of Moderna, the exact same vaccine last week. We all got Moderna, and everybody had a different effect. One person came in and just had a swollen lymph node, no problems. I was basically in bed all day Saturday. I had a student who got it on Tuesday and had a fever until Friday. So there's a few things to consider, and I often talk about the fitness of your immune response with this, and I don't have time to go into this today, but there's a lot of things that make your immune system more or less fit. So the example I give about myself is that I did not go to daycare as a child, so I was not exposed to a lot of the bugs that my daughter is getting now, and so I am like constantly sick when she comes home with something, constantly. Whereas my husband went to daycare. His mom actually ran a daycare, and he never gets sick to anything. So I just think that his immunity was built up better when he was younger. And he's also younger than me too, so that probably helps. But I don't think that has anything to do with it. You're still getting your protection. You're still getting your immune response. Okay, so it has a lot to do with just in general how your body reacts to vaccines or to what's in the vaccine. So in particular, if they had a certain adjuvant in there that just didn't really, you know, I'm not saying for COVID, but in general, let's say you just have a more adverse effect to like a flu vaccine. It might be that whatever adjuvant's in there, your body just doesn't like. And then it says, why are most vaccines injections? Would there be any advantage to a nasal vaccine for respiratory disease? Okay, we talked about that. So, you know, I'm going to talk a little bit about mode of vaccination. So let's hold on that one because I have a couple slides on that. And then I'll take these two and then I'll keep going. After initial infection or vaccination and priming of the immune system, subsequent infection causes the immune system to go into action as your graph showed. What parameters, factors, etc., determine the nature and the strength of subsequent responses vis-a-vis the wearing off of immunity and need for boosters? Yeah, this is great. I think that this is studied in real time is the bottom line. So that's kind of what they're studying now in these patients. And you know, for me especially, I'm interested in what happens, you know, when the person gets vaccinated, but then also what happens, you know, 6 months from there and then a year from there. So actually, we have this study, this aging study that I'm really excited to start after the pandemic lifts a bit. It's called the RIGHT trial. And we're going to look at the fitness of the immune response in patients over 70, actually, healthy people, not cancer. And we know these individuals have a weakened immune system. And then we're going to the plan was to give them the flu vaccine, right? And then see how their flu responses were based off of if they had a better immune response or not as good of one in that patient, in that demographic, because that's kind of when we start to see your immune system start to kind of go down a bit and not be as great. And so what we're doing with that is that we were going to use the flu vaccine, but I'm really hoping we can use the COVID vaccine because that's something that would be really interesting. So I think the jury's still out. They figured it out for some, like they give flu vaccine every year and they tweak it a bit depending on what type of variants are present, but they don't get it 100% correct. There's still people that get the flu, right? So there's a lot of different variants out there. And so we just have to protect ourselves with the most common ones and then go from there. Okay, and then the last one I will say, if vaccine limitations continue and second dose is delayed beyond 42 days, what is the next step? Start over with 2 or just get 1 more of the same vaccine or any available vaccine? The recommendation currently is to start over. I think that the jury's still out with COVID anyway. You know, I have been telling people that, my experience with people that have gotten Pfizer versus Moderna, it's been okay to schedule and it's been within the time frame, so this really hasn't been an issue. The Moderna has seemed to have worse side effects, so that's just something to look out for. But the reason I really like that the J&J shot is coming online is because it really is going to allow for people to just get one shot and not have to go back. But I think the jury's still out on that. I don't know. I mean, technically, you should have protection even after the first shot, right? So if you talk to an immunologist, they're going to say it doesn't really matter when you get your booster because you're just going to reinvigorate your immune response. If you talk to people that make vaccines, they're actually studying this in real time. So I think we'll know more as these studies come out. Okay, I'm going to stop there, and I'm going to just go through a few more slides because I do want to cover just a little bit more before I take any other questions, because I did have a few extra slides on vaccines as well. Okay, so as you were listening to the answers of the questions, hopefully you're also taking a look at this schematic that was put together by my colleague at Yale. Actually, she published it and shared it on her Twitter page, basically showing this is what happens with adaptive immunity. And so when you ask about how do we educate, you can use tools like this. This is why immunologists are trying to make tools for people to educate individuals about vaccination and immunity and what happens when your immune system is educated, okay? So these are all the things that I showed you. This is dendritic cells picking up virus, activation of the dendritic cell, and then migration to that lymph node, which is going to have, similar to tonsil, these nice centers where you can have this education. And then, educate T cells and B cells, and then they go on their way and they do their job, okay? So the question is, what happens with COVID-19 patients? So I've just showed you these beautiful images, which are similar to the ones on the right-hand side. These are patients that don't have COVID on the right. You see these nice little centers here where you can see these education centers where the immune system is getting educated in the lymph node. So what we do know about COVID-19 patients, which is what you can see with this image on the left-hand side, is that these germinal centers are kind of in disarray. You don't really have the robust kind of nice, neat, tight zones that you're seeing here with these images. You're seeing kind of some of that happening, but not great. And so this is one thing that we do know about the virus. So unlike something like the flu, where your lymph nodes are completely fine, it looks as though this virus in some individuals, not everybody, and this is later, this is somebody that has very severe disease, in fact, that they actually don't have as robust education centers in their lymph node. And so this is something that we've been thinking about a lot. And I think what I just want to point out here is if you hear about this, it's not that you're not getting the immunity that you need. It's just that it might be happening in a different way. And so especially B cells can be educated outside of these centers and can produce antibodies without problem. However, when that happens, they tend to be shorter-lived responses than longer-lived responses. So this is why we're actively studying this. I'm actually super excited about this because part of what my research program does is look inside tumors of patients and look at these structures and look at what's happening in patient tumors as a result. And so I think I'm going to get an opportunity to kind of study this a bit with COVID patients because it parallels nicely with what I'm already doing. And so this is how that effective immunity occurs, like I mentioned, in a dendritic cell, right? When you're getting that priming in that perfect world. And then you can get that innate and then adaptive immune response, which also includes antibody. And so my point is that we can get antibodies other ways. So even though we're still trying to understand in patients that have severe disease, what causes that disaggregation of their lymph nodes, we still think that they could have protective immunity. It just might come about a different way. And that's an active area of investigation. Okay, so in the last few minutes before I take some more questions and open it up further for questions, I want to talk a bit about vaccines and what we would call otherwise known as a biotherapeutic, right? So these are the considerations we have for vaccines, and this kind of hints at some of the questions that were asked in the chat box. So prophylactic versus therapeutic. So prophylactic means you give it, in anticipation. Therapeutic means you're treating what's there. So prophylactic is what we do with chickenpox, what we do with flu, what we do with COVID-19 now. Therapeutic is more what we do with cancer. It's very hard to educate our immune system beforehand for cancer because there's multiple targets, and that's a big business in cancer, trying to figure out what the best targets are for a vaccination, and it's an active area investigation. But I would say that it's much more difficult than something like a flu or even COVID. The second is mode of delivery, so systemic versus site-specific. So we know that a lot of the vaccines that we're getting from for viruses are systemic, and they seem to be pretty effective, barring some of the things I mentioned earlier. However, there are opportunities for site-specific vaccination, particularly for cancer, and that's something that we're trying to do. But that's also why you often see, you can get your flu vaccine intranasally versus intradermally. So I do have hope that eventually we might be able to have site-specific vaccines for something like COVID, but I think that we're just trying to kind of get as much of the population vaccinated as possible, and then can kind of develop some of that downstream. And then the last thing is specificity, which we've already talked about a lot. You need that adaptive immunity, you need that recognition, you need that education in order for it to work appropriately. So I think the best proof of principle of this, actually in the population, which kind of bridges virus and cancer, is HPV vaccination. So HPV vaccination was it was unclear actually when I was in graduate school and in my mid-20s if it was going to be effective or not. And at that time, they were vaccinating people, and I actually ended up getting my vaccinations. But what's been quite clear is that it's pretty much 100% efficacy. I mean, it's done really, really well. And so this is a situation where a cancer is caused by a virus. 90% of people that get cervical cancer have had HPV, right? Like you can have HPV and not get cancer, 100%. But if you have HPV and you look at cervical cancer patients, it's most of the time driven by HPV. And so because of that, we knew what the viral protein was. We knew what to vaccinate against. And so we did it. And it worked really, really well. And actually, there was a follow-up. This was in 2014, but it's even better now, showing that there's long-term efficacy and safety with the vaccine. It's very immunogenic. And now they're even treating not only women who are obviously going to be the most affected by it because they're going to get cervical cancer as a result, potentially, if they are HPV infected, not always, but potentially. But men are the carriers. They're the ones that transmit it. So they also have had a lot of conversations about also vaccinating males as a result. So this is one example of something that's been effective, but it's also still developing, essentially. Okay, so let's talk a little bit about, just take one step back about basic biology for vaccines, because I want to talk about the different types of vaccines that are coming out, particularly in relation to COVID. So you have that information. So central dogma for biology is that you have DNA, which is double-stranded. You can see it here. That's then transcribed into mRNA or RNA. And then that RNA codes for polypeptides, which then make a protein, and that's translation. So this is biology 101, okay? So the vaccine platforms that are out there can use either DNA or RNA, or can use protein, or it can be subunit, right? So these are all different types of vaccines. Okay, so if we go down here, this is mRNA. So the reason that mRNA is attractive is it can be rapidly developed, and it's a very low cost to manufacture. So this is what was developed to start with. The other reason it was very attractive for COVID is that the backbones that they wanted to use were already developed and proven to be effective, and that was a big part of getting it out quickly, right? So that goes back to me saying, like, it was tested, it was shown that these delivery methods were okay and safe, okay? And so this kind of disadvantage point, I don't 100% agree with, because I do think that there's been a lot of mRNA backbones that have actually worked really, really well. And so the way that works is that you get the RNA, and per dogma, it comes into the cell, and then that cell's machinery makes the viral protein, and then the viral protein can be basically released, and then those APCs that I mentioned to you can pick them up, and then they can educate your immune system essentially, and then start to kind of have this loop of education. So this is what we talk about when we say spike protein, right? That's a spike protein that is made from the viral mRNA, and then that way the APC can then educate all arms of the adaptive immune response, both killer T cells as well as helper T cells as well as B cells. So it's a very effective way to basically get virus made using the machinery within the individual. the vector vaccines, which is like an adenovirus vaccine, is basically going to do a similar thing. It's just going to package it essentially with like a Trojan horse virus, so that then your body can see that as non-self, take it up, and it's not going to replicate that Trojan horse virus. It's just so that your immune system sees it, and it's going to make more and then go from there. And so these are the different types of corona vaccine that in development: protein-based, viral vector, and mRNA. Okay, so this is what I just mentioned to you, mRNA, that codes for this spike protein, that your body does the work in that sense. It makes the spike protein, and then your education for your immune response occurs based off of what I just mentioned. Adenoviral vector, this is your Trojan horse, right? That basically it packages the spike protein, and then your body then produces more spike protein, and then your immune system gets educated. And then the protein base is just spike protein is purified and injected. This is by far the most expensive. Okay, so this is kind of the three different types that are in development, and what a lot of the population is getting now are these mRNA vaccines. Okay, so the other thing I want to mention is understanding the virus, and I just want to remind people of this, is that the reason we're all actively studying this is that making better therapies means understanding the virus. So, you know, the observation that I shared with you, that the immune response in those lymph nodes and those COVID-19 patients that have severe disease, they just aren't as well formed. And so that's something that would be an obstacle, and we need to learn that. So that's why all this research is occurring. Again, as I mentioned, knowing the R naught, which we talked about, so it's higher for COVID-19. Incubation time, this is a really tricky one for COVID. It's 1 to 14 days. And honestly, like the time of onset of symptoms is just all over the place. And this is why also we've had a lot of problems with asymptomatic transfer, because of the amount of time the virus stays around, and then people don't get symptoms. And so they're you know, basically spreading it to each other. Obviously, we know the hospitalization rate and the fatality rate is increased compared to flu. So we're learning all of these things to then, you know, educate ourselves. Okay, so what I want to share with you as kind of a last bit, because I think given the time, we won't have time to really go into cancer, but I'm happy to also provide my slides, Brad, if people want them. And you can kind of go through them yourself. I think they're pretty self-explanatory, and we've kind of laid the foundation. But one thing I do want to mention is, you know, one thing we're starting to understand better about the COVID-19 virus is viral entry and side effects. So as I mentioned, you know, we harbor it in our nasal passages, but the reason that you're feeling some of these weird symptoms that you feel, like loss of taste, for example, or loss of smell, is where the places that can be infected are present in your body. So ACE2 is the receptor that's expressed on multiple cells throughout your body in some parts of your brain. Okay, and so that ACE2 is going to be how the virus gets in to the cells. And so as a result, this can explain these non-straightforward side effects that we see from COVID-19. Okay, there's been a whole gamut of side effects, and that has to do with where the virus can enter. Okay, and ACE2 is one of the receptors that allow entry. So one thing that we did with our local study, as Brad mentioned, we did a local study on patients in Pittsburgh that were really that did really well, that entered the ICU, and we studied patients that came out of the ICU and recovered. We studied patients that died as a result, unfortunately. And so what we did is we actually looked at ACE2 expression also on immune cells, because we wanted to understand if they were able to be directly infected, because you could imagine if the virus then can get into the immune cells, it's going to also affect those immune cells. Similar to, akin to like HIV, one of the things that HIV does is it infects CD4 helper T cells. And then therefore, if you don't have a lot of those, then your CD8 T cells don't function very well. It's a whole kind of waterfall effect. And so what we actually saw is that gray is a healthy donor. So age match, somebody that's, you know, probably between the ages of 45 and 70 is what we chose for this, because that was what our patient population looked like for the most part. Really no ACE2 expression here. But in COVID-19 patients, we actually saw elevated expression on the monocyte population. And the reason this is important is these are the precursors for those antigen presenting cells that I mentioned to you. So they're going to turn into a dendritic cell or a macrophage potentially within tissues. And so these can be directly infected. And we know that this is severely upregulated on COVID-19 patients that you can see here in this bar graph. But in patients that have non-COVID ARDS, so basically just have lung disease that they could potentially pass away from, which is one of the manifestations of COVID as well. But these patients just don't have COVID, they just have ARDS. It's not as high. So we know that this is something that is happening systemically. Okay, so this isn't even at the site. We also looked at the site. We did endotracheal brushing or washes, basically, which allows us to look at like, nasal passages, etc. And so we haven't had a chance to do that because we had limited cell numbers. But what we did find was that in the peripheral blood, even the virus is infecting these cells. Okay, so one thing I want to give props to is that one effort that is being made at Pitt, and this is just one of several, is a very multi-team collaborative effort to study the immune response to COVID-19 in patients that are ill or critically ill in the ICU. And these are all the people that are involved. I mean, it's a huge effort. I'm scientifically leading these efforts with Daria Vinali, and we have a very talented team of people that are helping. But really the patients and their families that are allowing us to take these patient samples and consenting with the COVID team that are boots on the ground and really consenting these patients and giving their specimens so we can understand the virus more. Okay, so with that, I'm going to stop, I think, at this slide. And I want to kind of go through this a little slowly because this is also something from our study that is currently actually out for publication, hopefully soon. We're waiting for it to get reviewed. But basically, this was captained by a very talented postdoc that Dario and I supervise. And basically, we did not only for these experiments, we basically did a technology called single cell RNA sequencing. So what that does is it allows us to look inside the cell and understand what is different about patients that died in the ICU versus those that survived. And even if you don't read these graphs well, one thing that you can see is that those that survived are all the way on the right and those that didn't are on the left and vice versa. Okay, so what we started to see is in those cells that I mentioned to you that are that get infected, the monocytes, we saw these programs, which we can see with the transcriptome. We saw these programs that were enhanced in patients that survived versus those that that didn't. And those programs are directly linked with how much cytokine is in their plasma. So we were able to kind of come up with this multivariate analysis that showed us, okay, individuals that had this program in their monocytes and this cytokine, they died more. And then patients that had this monocyte signature and this cytokine in their plasma, they survived better. And that also is shown here with survival curves, right? So if you have high and low, what happens? This patient here is surviving, these patients here are surviving better, where these patients are not, and vice versa. So this is just one of the insights that have come from our studies, which I'm really excited about, because I think that we're really starting to understand the immune response. And if you understand the immune response, then it helps us combat it even better if we have to deal with variants or we have to, it's not going to go away necessarily. I think that's the common misconception. So this is a really huge research effort that I'm really proud of. Okay. And so I'll end with this and say, so we have a vaccine, what else should we worry about? And so this goes back to the idea of antigenic variation, basically. So we have to worry about our immune system being evaded. And the way that that happens is called with antigenic variation, which is what is being talked about now. So antigenic drift is a point mutation in the viral sequence that basically is going to just cause evasion of the antibody that's going to neutralize it, right? It's not that horrific, right? Antigenic shift is a whole reassortment of the segmented RNA. So it looks completely different than the initial virus. And this is what causes, not like we're in a pandemic, but this is what causes like a raging epidemic, okay? And also how quickly it mutates, which is also something we don't know very well because we don't know if these are all the variations we're going to see. So this is something that we're keeping in mind as we're studying this. And these are the two terms that we use to define that. So I'll leave it here and say, we have to worry about just this evasion, right? So we have this vaccine and that's great. But, we know from HIV, for example, that it can evade the immune response. It can kill CD4 T cells, as I mentioned. It can infect those immune cells directly. We know also that the HIV virus rapidly mutates, right? So these are the things we have to learn in order to understand how to combat it better. And the same with cancer. We didn't really get to talk a lot about cancer tonight because I want to leave time for some additional questions. But cancer is the same way. This is a real challenge for us. We have even more immune evasion mechanisms that we're still trying to understand in cancer patients that are very similar to what happened with virus and some that are unique. And we're trying to target those, and we're trying to, regulate the immune response in those patients to be better as a result. Okay, so with that, I'll leave it on this slide, and I will open up my Q&A again, and kind of just maybe pick a couple of questions here. So vaccines and autoimmune disease, should people with autoimmune disease get vaccinated in general? Again, I think the jury is still out here. Of course, if you have an autoimmune disease that is going to, so the reason, so let me take a step back. The reason we get autoimmunity is because we have too much of seeing self and its destruction of self, aberrantly, right? And so it really depends on the type of autoimmune disease you have. I know a lot of people that have had autoimmune diseases, is what I can say, and have been recommended by their doctor to actually get vaccinated. And so I don't think you should be deterred, but I would definitely, because there's so many different autoimmune diseases and we don't have time to talk about all of them, I would definitely consult with your doctor or your physician and ask them. But in general, individuals that I know that have autoimmunity actually have gotten vaccinated. Is there a difference between variant and strain? Same idea. So same idea there. What do you know about the prophylactic vaccine that Moderna is working on? Sorry to disappoint you, Carol. I don't know anything at this point. I think that it's a work in progress. I think there's still some things that we're trying to learn. I keep hearing the phrase chronic inflammation and how this allegedly causes from severe disease to, disease and depression. Chronic inflammation, a real phenomenon. So chronic inflammation is not necessarily a good thing, right? That's what we get when we get autoimmune disease. You actually have too much. I often talk about the Goldilocks effect with the immune response. You want to have something right in the middle to keep your body at homeostasis. You have too much, it's not good. If you have too little, it's not good. So you have to be somewhere in between. So autoimmunity is like the too much, and the other side of the pendulum is too little, especially if your immune system's getting evaded. So it's not usually a good thing. The other thing that comes with we are seeing with COVID patients is cytokine storm, which we've talked a lot about in the OSHA class that I'm helping out with. And cytokine storm actually is also not a good thing because it causes a lot of tissue damage. And so there's been patients that have had really very few symptoms of COVID and then had severe cytokine storm in their lungs and had a lot of tissue damage. So in general, chronic inflammation is tough. Reducing it, there's a lot of things, that they would recommend for natural lifestyle, fitness, diet, things like that. But in general, that's something that you also, depending on the type of chronic inflammation you have, you would talk to your physician about. It seems vascular system compromises are a fairly significant post-COVID risk factor. Is there anything one should do to minimize the risk? It's a good question. I actually don't think I know the direct answer to this. I would say that I guess it depends on what you mean about vascular system compromise. Maybe circulation is what you're referring to there. We can kind of circle back to that if you want to clarify, Anne. Okay, let me see. We already talked about that. Oh, and then I missed one. Can you explain how immunity is or is not transferred through breast milk? There must be an evolutionary reason that antibodies are present in breast milk, but vaccination immunity doesn't transfer this way. Are antibodies completely broken down by stomach acid? Basically, how much protection would a breastfeeding baby get from their COVID vaccinated mom? So the bottom line is that, and I am definitely not an expert in maternal fetal transfer. Actually, there is an expert in the Department of Immunology at Pitt. If you want to invite him to give a seminar on that, his name's Tim Hand. So Brad, I can give you his contact information. That's what his whole research program is on. But what I will tell you about breast milk is that it's really just the bare minimum to get your baby by until they start to develop their own immunity. Okay, so that's it. That's the long and short of it. You're getting very, very minimal protection, but necessary protection while your child develops that immunity. And so in fact, like when I had my daughter, I was pretty adamant about breastfeeding, at least for the first 6 months, because I knew that she was going to be developing her immune response and I wanted her to at least have some protection while she was developing it. And so, and then after about 8 months, my milk supply just wasn't there. And so I actually had to supplement with formula and she was completely fine. So they usually tell you that it's not a bad idea if you can swing it. But then there's also people that never breastfed and their kiddos are okay. So it does really help with just that baseline kind of immunity until your child starts to really develop their own. I have not seen that neurological manifestation for COVID. That has not been noted. I'm not sure what this means, Phyllis. Sjogren's disease with the vaccine. And, oh, developed vasculitis, blocked, delayed, and then died. Okay, so that could just be due to some sort. So I would be interested to see the empirical data, like if it was just because, you know, as I mentioned, there's a lot of odd side effects from this. So the ones that have been noted, quite frankly, that are common now, that are actually on the list, that weren't on the list before, are loss of taste and loss of smell. Some of these other things that you're mentioning with vascularization, this could just be due to those ACE receptors being present at a higher frequency in that tissue or in that area or in that vascularization, and then maybe causing deterioration as a result. So that seems like it might have to be considered on like a case-by-case basis. So what I would say is when you're educating yourself and you're reading or talking to people about like one-off cases about the vaccine or about their experience about being infected with COVID, I would definitely say that you should streamline and think about what has been shown empirically in a larger group of individuals, right? That's how we get our data as scientists. We do have case studies and you do have things that happen that are odd, right? But I would say that if you're educating yourself, I would use the facts from the actual data that are there from a large group of people, right? But my guess on that would be for the vasculitis would be where the ACE2 was higher, because that's the other thing we don't know. We know ACE2 is on all of those tissues, as I mentioned, but we don't know if some people have more in certain areas than others, because we know from the immune response perspective that we see patients that have more versus less. And so that's kind of where we have what we have for now. Okay. With Sjogren's being an immune disease, is there a reason it might not be effective with the vaccine? I don't think so. I don't have any reason to think that that would affect it. But again, these are all things that are evolving. So what I can educate you on is why vaccines are important, what we know now, where to get your advice, where to understand about the immune response. These are all little kind of nuanced. So we have to just wait essentially and see what happens. I'm just reading, sorry. Okay, so basically what's known about the long hauler effect, basically if it stays on, right? And so maybe the immune system saying revved up and overactive than needed. That's possible. The mRNA could potentially cause something like that because of the way in which the protein's made, and then you're having this kind of cyclical effect. But in general, your immune, we didn't get to talk about this, but in general, your immune system has a homeostatic point, right? There's homeostasis. So you have cells in your body that shut down the immune response, or receptors that go up on those killer T cells to tell them to stop. And in general, that works for most people. I would say that if they're seeing that sort of a reaction, then that would be something then to consider downstream if it was happening to enough people. And that has to do with the dosage, right? So how much is given? So again, back to this Goldilocks effect. So I don't know that enough data is out for enough individuals to say, yes, this is going to be a definite problem. But I would say that it will be out soon, and we can then say, okay, this is now too much of an immune response and we need to dial it back, or it's not enough and we need to give more. Okay, so I'm gonna stop there. It looks like I have answered most questions. Kathy, we unfortunately do not have time to go into the HIV vaccines, but definitely email me. I can send you some literature. What I will say about HIV is that it's actually a very, very manageable disease, quite frankly. At this point, we know a lot of what happens, and even with it varying quite a bit. A lot of people that are on antiretrovirals do quite well. And really what I've learned from my colleagues that really actively work in HIV, I used to a long time ago, but not for a long time, is that if you're really good about taking your meds, and taking those antiretrovirals, it's very, very manageable. As far as vaccines go, I can send you some literature, and there are some, thoughts around that. But I think that because the antiretroviral therapy works quite well in most patients, that's kind of the direction that that people go. Okay. So as I mentioned, I had some other slides. I apologize that I didn't get to go through everything. I thought we did a pretty good job. It was a pretty large tour de force. You can see that I had multiple slides that I put together in anticipation. But yeah, so I think I will say that I'll end here. If there's no other questions, I will stop and say, please email me if there's something really burning we didn't get to. Brad, I'm happy to also share my slides with anybody that's interested. Totally okay. And I'm really happy that I got to do this. I really love doing things like this. I love the energy and enthusiasm and the questions that are thrown at me. It challenges me. It makes me think harder about things that I need to know to educate people better. So it helps me build kind of my library up. But thank you so much for your time and attention. I super appreciate it.
And thank you so much for being here. And one thing I love about the Café Sci audience is the questions that they come up with. So it is
They do.
a
They
wonderful,
come up with,
thoughtful group.
yeah, they're a very thoughtful group. They remind me a lot of my OSHA class, which has been really, really wonderful to teach as well. So I'm really excited that I've gotten to do this, and maybe I can come back and talk more about cancer next time. I just thought we should talk about immunity and COVID because that's what's happening right now. So definitely let me know if there's anything else you need.
Yeah, I appreciate the timeliness of your talk tonight. So thank you so much for it. I would be remiss if I didn't thank you all for tuning in and joining us this evening. And if you're interested in supporting Café Sci, of course, there's always the option to throw a big bag of money at us, and we'd take that no problem. However, one of the best things you can do to help Café Sci is to spread the word and invite friends and family members and acquaintances and your third grade teacher to join us. Next month's speaker will be Dr. Jennifer Sheridan from Carnegie Museum of Natural History. She works as an assistant curator of amphibians and reptiles, and her field work is with tropical frogs in Southeast Asia. So it's going to be a fantastic presentation as well. So some fun things happening there. And as Dr. Bruno said, if you wanted to get those slides, you can email us. There's an address on the Café Sci page at carnegiesciencecenter.org, and we'll get in touch. And you can also check this out again on YouTube and share it with friends shortly after we're done tonight. Tullia, thank you so much for being with us. I really appreciate it.
Thank you. Thanks for having me. Thank you, everybody, for the thoughtful conversation and questions. I really appreciate it.
All right. Have a wonderful night, everybody. Thank you. We'll see you in a month.
Bye-bye.